Hi everyone, welcome back to my channel Pharmagist. Myself Dr. Haimavati. In today's video we will learn about penicillins. So what are penicillins? So penicillins are a class of antibiotics. So whenever you go to doctor, so suppose you are suffering from fever or any infection, whenever you go to a doctor, so the most commonly prescribed antibiotic is penicillin. So penicillins, the discovery of penicillins is also a very interesting topic. So now let us study about penicillins, how they were discovered and what is their different aspects of penicillins we will learn in this video. The penicillins, they were discovered by Sir Alexander Fleming. So Alexander Fleming was a great scientist. So he dedicated his entire lifetime for identifying different drugs so that they will help human beings to have a better quality of life and improved health conditions. So he was conducting experiment on some bacteria known as Staphylococcus. From that he identified the penicillins. So this picture I got from the internet. You can see here the picture. So in this you can see the mold which makes penicillin. So he described this mold which he observed during his experiment as the mold which makes penicillin. And this clipping also I got from the internet only. So it was written in this that Alexander Fleming, he devoted his entire lifetime to medicine. So he was studying a bacteria called Staphylococcus, which was a flu virus. And then when once while doing the experiment, he noticed that a mold was formed on this Staphylococcus bacteria, and he observed that wherever this mold was formed, there the bacteria is killed. So he identified a zone of inhibition on the bacteria. So thus he identified that this mold is killing the bacteria. That means this mold has some property which is killing this Staphylococcus bacteria. And later he realized that this mold was an antibiotic, and he named that antibiotic as penicillin. And the penicillin, it was discovered in the 1930s, and the bulk synthesis was carried out in the 1940s, and it was during the World War II. And this identification of this penicillin, it helped the life of millions of peoples in those days. It improved their quality of life, and during the World War only, so a number of soldiers were injured, and the identification of this penicillin helped cure those soldiers, and this was a major landmark in the history of medicine. So for this he was awarded the Nobel Prize also. Now let us see the different aspects of penicillin. So as I said, so he identified that this mold was growing and it inhibited the Staphylococcus bacteria. Now as per our BPharm syllabus, what are the different things that we have to study? The first is classification, nomenclature, stereochemistry, mechanism of action, and structure activity relationship. Now coming to the classification of penicillins, there are a number of classifications for penicillin. We can write according to our convenience and understanding. So I have I have gone through all the different classifications, and I have combined all the classifications into one thing. So we have narrow spectrum, broad spectrum penicillins. Narrow spectrum means if they are acting only on one kind of bacteria, they are called narrow spectrum antibiotics. And if they are acting on more than one type of bacteria, for example, if they are acting on gram negative and gram positive bacteria and a number of other bacteria, that means they are covering different classes of bacteria, they are called broad spectrum antibiotics. And natural and semi-synthetic, so based on the source of obtaining, how they were obtained. So if they are obtained from naturally, they are called natural synthetics. That means the fermentation of this penicillium mold. So fermentation of this mold called Penicillium chrysogenum gives the basic extract of penicillins. So they are called natural penicillins. So we have penicillin G and penicillin V. Only these two are the natural penicillins. And then we have semi-synthetic penicillins. Semi-synthetic penicillins means they are combined. So the fermentation extract from natural process is taken and further different chemicals are added in order to obtain more complex structures of penicillin, which are resistant to the penicillinase enzyme, which are resistant to the acid enzyme, etc. So they are known as semi-synthetic penicillins. So in the semi-synthetic, again we have penicillinase sensitive broad spectrum antibiotics, and again in that oral or parenteral, that means by the route of administration. If they are given orally, they are oral semi-synthetic penicillinase sensitive penicillins. If they are given parenterally, that means through injection, if they are given, it is called semi-synthetic penicillinase sensitive. Next we have semi-synthetic penicillinase resistant oral or parenteral. So here we have penicillinase sensitive and here penicillinase resistant. So this is an overall classification which covers all the different classes of penicillins. Narrow spectrum, broad spectrum, natural, semi-synthetic, and in semi-synthetic we have penicillinase sensitive and resistant, and again broad spectrum. Okay. Now let us see the structure of penicillins. So the basic structure of penicillins is the cyclic cyclonamide structure. So when the cyclonamide structure is cyclized, you get this four-membered ring which is known as beta-lactam ring. So this beta-lactam ring is the main pharmacophore of penicillins, and it is also known as a monobactam ring or azitidinone ring. And if this beta-lactam ring, if it is attached to a 5-membered thiazolidine ring, it is known as penam structure. And if this penam structure is having a double bond at this position, for example, if we give the numbering from here, okay, so this is 1, this is 2, this is 3, and this is 4. Okay, so if this penam structure, if we have a double bond between this second position and third position, it is called peneme ring. And then if this 5-membered ring is replaced by a 6-membered ring, it is called cepham ring. And in this cepham ring, if it contains a double bond, it is called cepheme ring. So double bond means E, alkene. So penam ring, peneme ring, cepham ring, and cepheme ring. So 5-membered thiazolidine ring means it is penam ring, and 6-membered ring means cepham ring. So here you can see when there is 5-membered ring, they are called penicillins. So this beta-lactam ring, if it is attached to 5-membered ring, they are penicillins, and we have different substitutions. And then if it is having a 6-membered ring, it is called cephalosporins. So penicillins and cephalosporins both have the beta-lactam ring, and 5-membered ring structures are called penicillins and 6-membered structures are called cephalosporins. So under the beta-lactam antibiotics, we have penicillins and cephalosporins. Now coming to the mechanism of action. So penicillins, they inhibit the cell wall formation of the bacteria. So bacteria, they have a cell wall surrounded. Okay, so the bacterial structure contains a cell wall. So this cell wall, what is the function of this cell wall? So the cell wall protects the bacteria from the external environment. So it is essential for the survival of the bacteria. If this cell wall is not present, what happens? It will be easily digested by the host. So if they are present in any organism, host, for example, if they are present in our human body only. So when this bacteria, if it enters into our human body, if this bacterial membrane is not there, if this protective layer is not there, what happens? The immune system in our body, it will digest this cell wall. This will digest this membrane bacteria. As a result, what happens? The bacteria will be killed. So that is the function of the cell wall of the bacteria. And this cell wall of the bacteria, it is having a polysaccharide structure. And again in this polysaccharide structure we have alternating units of NAM and NAG. So here you can see more detailedly. So there are different polypeptides which are present, and this NAM, N-acetylmuramic acid, and then NAG, N-acetyl-glucosamine. So these amino acids, they are arranged alternate to each other, and here you can see cross-linking of these peptides takes place, and this cross-linking is brought about by transpeptidase enzyme. So this transpeptidase enzyme is a penicillin binding protein. So this penicillin binding protein, there are three types of peptidases, and we have transpeptidase. So these penicillin binding proteins, they contain carboxypeptidases, endopeptidases, and transpeptidases. So these three together, they are known as penicillin binding proteins. And these penicillin binding proteins, they are responsible for the cross-linking of these peptides. If this cross-linking step is prevented, then we can prevent the formation of the cell wall of the bacteria. So here you can see all the beta-lactam antibiotics, what they do, they prevent this step cross-linking. So here you can see there is cross-linking, and when this beta-lactam antibiotics, they interfere with the binding of this polypeptide. So when they interfere with the binding of this polypeptide, what happens? So there will be no cell wall. If there is no cell wall, means the bacteria will ultimately die. So this is the mechanism of action of penicillins. Now let us see the stereochemistry of penicillins. Coming to the stereochemistry of the penicillins, we have to explain the structure of this penicillins in the three-dimensional space. That means how many atoms it contains, how many rings it contains, which atom will be above the plane, which atom will be below the plane. So that is the stereochemistry. Stereochemistry means it is nothing but the three-dimensional arrangement of the functional groups or atoms present in the molecule. So I have seen a number of students, they get feared by the name of stereochemistry itself. So they picturize stereochemistry as a very horrifying thing, but please don't be fearful like that. You should only understand that stereochemistry is nothing but three-dimensional picture of these penicillins. For example, consider if you want to describe a person. Okay, so any person of whom you know, or example, uh, the more easiest example will be, consider a heroine. For example, Deepika Padukone. How will you describe how Deepika Padukone looks if somebody doesn't know her? Then how you will tell? So you will tell her what is the height of Deepika Padukone, and then what is her weight, what is the skin color, and different things like that. So similarly, not only for penicillins, for any molecule, stereochemistry means it is nothing but the three-dimensional representation of the molecule. So how you will describe the molecule, how it when you see it. So here this penicillin, they contain this uh, PNAM structure. So PNAM structure means it is a bicyclic ring. So this is the first one and this is the second one. Bicyclic ring means the beta-lactam ring which is fused with the thiazolidine ring. So that is the first point. And next, when you give the numbering, so the numbering starts from nitrogen. So according to USP, the numbering starts from nitrogen. So first let us give the numbering. So it is containing 7 atoms. So the first point in the stereochemistry is it is having a bicyclic fused structure, and the total number of atoms are 7. And at second position, what is there? The carboxylic acid group is there. Okay, so second position you have carboxylic acid group, and here the carboxylic acid group is given with dashed lines. Dashed lines means it is below the plane. So this carboxylic acid group at position 2 is present below the plane. And next, At third position we have two methyl groups, they are in the plane. So this normal lines they represent that they are in the plane. Dashed lines represent that the molecule is below the plane. And this solid line here at sixth position you can see, this solid line represent that it is above the plane. And next at position 5 and 6 we have two hydrogens. So these two hydrogens also they are uh, below the plane. So in these hydrogens also we have dashed lines. So in this figure it is not there, so I am drawing the dashed lines. So hydrogens at position 5 and 6, these two hydrogens they are below the plane. Okay, so third point is these hydrogens are below the plane and they are in cis arrangement. So both the hydrogens, because both of them are below the plane and both are, that means they are in the same plane and they are near to each other. So these both hydrogens are neighbors. So because they are neighbors, they are cis to each other. And at next point, fourth point is the at position 6 we have this amino group, and this amino group is above the plane because it is a solid line, it is above the plane. And next fifth point is this position 2 and position 6. So this carboxylic acid group at position 2 and amino group at position 6, they are trans to each other. Okay, so this carboxylic acid, it is having dashed lines, so it is below the plane, and this amino group is a solid line, that means it is above the plane. So one is below the plane and one is above the plane, so they are opposite to each other, that means trans to each other. Okay, so simply this is the stereochemistry of penicillins. I hope you understood. And next let us see about the structure activity relationship of penicillins. Now coming to the structure activity relationship, so the first point in this is the beta lactam ring is essential for activity. Okay, so the basic pharmacophore of this penicillins. So pharmacophore means the functional moiety or the molecule which is responsible for the activity of penicillins is known as pharmacophore. So the basic pharmacophore, that is the beta lactam pinam ring. So the 4-membered ring which is fused with this 5-membered ring. So this pinam ring is essential for activity. So that is the first point in the SAR. And the next, presence of this carboxylic group at position 2. So this we have at position 2 carboxylic acid group. So this is essential for activity. And also the nitrogen at position 1 is also essential for activity. And at position 3 you have two methyl groups. So at position 3 we have this one and this one. These two methyl groups, they are also essential for activity. Now next, fifth point. So at position 4 we have sulfur group. So this sulfur is also essential for activity. And if you oxidize this sulfur to sulfoxide, what happens? The activity is reduced. Activity is reduced and the acid stability is increased. So in this case what happens, if you oxidize the sulfur, the stability of the penicillins will increase, but at the same time the activity will be reduced. And then at position 5 we have hydrogen. So this hydrogen you cannot replace with any other functional group. So presence of this hydrogen is essential for activity. So that is another important point. And next, at position 6 we have this amino group, and this amino group again it is attached to an R group. So this presence of R group also is essential for activity, and this R group you can substitute with any alkyl groups or bulky groups, so that also improves the activity. And again at position 7 what we have, this carbonyl oxygen, and this carbonyl oxygen is also essential for the activity of penicillins. So overall what does it indicate? If you remember the penam structure of this penicillins, you can easily answer this question. Okay, so penicillins, So this structure is very important to understand the stereochemistry as well as the structure activity relationship. So if you basically remember this structure, you can easily write about its stereochemistry as well as the structure activity relationship. So you can see that each and every element in this penicillin structure influences greatly the activity of penicillins. So the presence of nitrogen at position 1, carboxylic acid group at position 2, the 2 methyl groups at position 3, the sulfur at position 4, hydrogen at position 5, and amino group at position 6, and the carbonyl group at position 7. So all these elements are essential for the activity of the penicillins. So they influence a lot in the activity of penicillins. Now coming to the uses of penicillins. So penicillins are used in a number of infections like pharyngitis, tonsillitis, scarlet fever, endocarditis, pneumococcal infections, staphylococcal infections, diphtheria, anthrax, syphilis, etc. So they have their uses in a number of infections. And side effects. So they also have a number of side effects like hypersensitivity, rashes, nausea, neurotoxicity. So these are the common side effects of all the penicillins. And now let us see the different structures. So as I already told you, at this position 6, this R group can be substituted with a number of functional groups. So when we are giving different functional groups, you will get different penicillins. For example, here we have penicillin G and penicillin V. This penicillin G and penicillin V are natural penicillins. So this is benzyl penicillin and this is phenoxymethyl penicillin. These two are the natural penicillins, and the remaining all these penicillins, they are semi-synthetic penicillins. So this R group is substituted with different bulky functional groups. As I already told in the structure activity relationship, that R group can be replaced with a number of bulky groups. So when you're replacing it with different groups, you will get here propacillin, ampicillin, amoxicillin, carbenicillin, methicillin, oxacillin, cloxacillin, okay, dicloxacillin. So these are the different penicillins. Maybe you have seen some of these names in your prescription. So whenever if you have visited doctor for any kind of infection or fever, so generally we see amoxicillin in most of the prescriptions. We also see oxacillin and dicloxacillin also. They are prescribed by a number of doctors. So when you see the prescription, try to remember this structure. Okay, so when you try to recall this structure, so practice these structures, draw these structures on your notebook, practice it several times, then you can remember it well. And don't be fearful that these structures are very complex. So the more you practice, the more you can remember these structures, and you can write well and get good marks in the examination. I hope you understood the video. If you like the video, so do not forget to like, share and subscribe. We will see in a next video. Till then, bye bye.